Hello, everyone. Can you guys hear me okay? If any of you can respond, if you can hear me okay, so I know that everything is fine. Can you see the, can you all see the screen? Yeah, I just want to make sure you can hear me okay. Oh, awesome. Okay, thank you. Okay, so I will turn off the webinar chat option and I'll give maybe a minute or two and then we're going to start. Yeah, maybe one more minute and then we're going to start. Okay, so we are as scheduled. We are 8:30 a.m. and we're going to start our seminar. Let me do this full screen. Okay, let's. Maybe at the end of the seminar, we have this Q&A session. So let's begin. Okay, what happened to my. Okay. I don't know why it got stuck, feels like. Okay, let's go with this one. Okay, welcome everyone. So my name is Shamli. I am with BioVision for about 3.5 years now. So I'm the Senior Product and Business Development Manager. And today I'm going to focus on all our in-house assay kits, focusing on click chemistry. So we have around 20 products in this category. So I'm going to give you the product highlights, some technical background, and why these kits are different and special, and why this makes these kits, this entire kits in this category makes it something special for BioVision. Here is an overview. I will talk about give you a general introduction about the click chemistry, what's the technical background, or what's the principle for all this category, and what are the application of click chemistry. And the different click chemistry kits available from BioVision, you can study either DNA synthesis or proliferation. You can study RNA synthesis, global protein synthesis, you can study apoptosis, and you can study phospholipid synthesis, as well as a lot of glycoproteins or other modification of proteins like fucosylation or meristylation or palmitylation. And finally, the technical specification I will go through for individual kits to give you an idea like how are we going to sell these kits to the customer. To introduce what's click chemistry. So click chemistry was the term coined by Sharpless in 1998 and it was fully described in 2001. So the term click chemistry, it actually refers to a group of reaction. It's not a specific reaction. As you will see in the subsequent slides, what do I mean by that? So it refers to a group of reactions, which is fast, simple, easy to purify, the final product. It can be applied over a wide range of different applications, and it's regio-specific, which means it gives only one special product. And overall, the yield is very high for the end product. So click chemistry, it's a method for we attach a probe or a substrate of interest to a specific biomolecule. That's the principle that we use, and it's called bioconjugation. And the readout can be either synthesis of DNA or protein or RNA. But the reaction that gives the output is click chemistry. And why it's important? So click chemistry has been used mostly in drug delivery and formulation study. So I will show you the applications of click chemistry in pharmaceutical sciences, why it's so important. So as I mentioned, what are the characteristics of a click reaction? It should be, number one, simple to use. It's fast. The final product will be easy to purify, and the yield of the product is very high. Application over a wide range of applications, and the byproduct that is produced in addition to the main product, there is no interference from that byproduct for detecting the main product, and it's homogeneous. It's stereospecific and regioselective, which means regioselective means using click reaction, it gives only one specific major product. So there is barely an interference from the byproduct that comes along with it. It's biorthogonal, which is the reactants and products that do not react with biomolecules or functional groups. And the reaction is thermodynamically favored, which means the reaction you can carry at room temperature. So all the experiments are carried at room temperature. You don't need special equipments or anything in terms of like the incubator or anything. And the solvents used in the reaction, it usually no solvent or uses a solvent that is benign or easily removed, like water. And it is very insensitive towards oxygen or water. So it's insensitive. So which makes click reaction very versatile, simple to use, and can be applied over a wide application. You don't have to be very careful about all the reaction conditions for these assay to work. So what's the application in pharmaceutical sciences? Drug discovery. It can be used in bioconjugation, and also, as you can see, as in radiochemistry. So let me give you an example in terms of like how it is used in drug discovery. So click chemistry has been used, just an example, that to produce the peroxisome proliferator activated, that is the PPAR at agonist for treating type 2 diabetes. So click reaction have been used here to develop this agonist. Another example is those in situ click chemistry have been used to identify HRV protease inhibitors. So you can see that click chemistry is very important in discovering all these agonists or inhibitors in drug discovery. Second is in bioconjugation. So bioconjugation means conjugation of your molecule to make functional molecule, biofunctional molecules. For developing these biofunctional molecules, click chemistry have been used. So what does bioconjugation mean? So it involves the attachment of the synthetic labels to biomolecular blocks, such as fusing 2 or more proteins or linking a carbohydrate to a peptide. And then we have the radiochemistry. So radiochemistry is like such as the the non-invasive nuclear imaging techniques such as PET scan, the positron emission tomography, or the There is another one it's called single photon emission computed tomography. So it has been used, click chemistry have been used mostly in the field of radiopharmacy, for radiopharmaceutical applications, and it has been very successful. It is an important tool to assess the novel drug candidates. That's what they study in radiochemistry. So you can see the application of this click reaction in pharmaceutical sciences. So now coming to, as I mentioned before, that click reaction basically refers to a different range of reaction, right? A group of reactions. So the four major click reactions are cycloadditions, nucleophilic ring openings, non-aldol carbonyl chemistry, or carbon multiple bond addition. But the one that we are majorly interested is the cycloadditions. That is the one that all our kits are based on this chemistry. So what's the chemistry? So cycloadditions, as I have written here, it's a copper catalyzed cycloaddition reaction. We say Huizen 1,3 dipolar cycloaddition, in short HDC. So we have a azide and a terminal alkyne. In presence of copper catalyzed by copper, it forms a 1,2,3 triazoles. So, so this is the most popular click reaction. So basically, all our kits are based on this cycloaddition click reaction, where you see the unsaturated reaction, azides and alkynes, they fuse. So the click reaction is unique because the azide moiety is actually absent in almost all naturally existing compounds. It lacks reactivity with natural biomolecules and consequently undergoes ligation only with a limited set of parameters. So you know that when you are using this reaction to see an output, there will be barely any interference from any third molecule because, as I said, the azide moiety is absent in almost all naturally existing compounds. So the popularity is attributable to the fulfillment of these criteria, as I mentioned, like the mild reaction condition, high yield, simple procedure, it's selective, and it's specific, and it's reliable. Okay, let's move this, and it's very easy to use. Okay, let's go to the next slide. I don't know why it's not going. Okay, so now coming to our BioVision's click chemistry based kits. So we have around 20 different kits. So what are the things you can study with? We have some kits like K946. It's for studying DNA synthesis and cell proliferation. If you want to study RNA synthesis, we have this K462. If you want to study protein synthesis, like the global protein synthesis, we have these kits. If you want to study apoptosis in your cells, you can, these are the kits. You want to study phospholipids, so mostly it's a phosphocholine based, which I will elaborately go through individual kits as well, more in details in the subsequent slides. If you want to study glycoprotein synthesis, as well as we do have for, the sialic acid, you can use these kits. If you want to study the modification of different proteins via myristylation or stereoylation or palmitylation, we have kits for that. These are the modification for proteins. Okay, as I said, one of the kit is for studying DNA synthesis and cell proliferation. So what's the principle? So typically all cells are based on, all the principle is we incorporation of labeled DNA precursors into the cellular DNA. So cellular DNA is during the S phase, when cells are actively dividing. That's when they are the proliferating cells. we incorporate this labeled DNA, and that's how you study the DNA proliferation. Okay, so classically, not click chemistry. Previously, what people have been using so far, using pyrimidine deoxynucleosides to label the DNA precursors. But this is autoradiography or like radioactivity, or it needs immunological detection using BRDU, bromodioxyuridine. But what are the disadvantages? As I said, thymidine, you need autoradiography. It's autoradiography based, which is radioactive. So it's low, it's time consuming, and radioactive is risky. Even the resolution of the images, it's a signal to noise ratio. Signal is low, noise is high. In the bromodioxyuridine method, it blocks access to anti-bromouridine, like when you use the antibody for the immunologic detection. So the bromodioxyuridine itself blocks the anti. And it needs denatured protocols. So that way, how click chemistry varies is, so here we have 5-ethynyl 2-deoxyuridine as the nucleoside analog. Instead of thymidine, which I showed in the previous one, you are using radiolabeled thymidine. Here you are using EDU as the nucleoside analog, and that get incorporated into the newly synthesized DNA. Once it gets incorporated, it stops into the newly synthesized DNA, and then this EDU, it has this alkyne group as shown here. And then you add the azide, which is the click chemistry. So we add the fluorescent azide. Then you have, it's a copper catalyzed. An alkyne group is coming from your EDU, the nucleotide analog. You are adding the azide in presence of copper catalyst. That's how the reaction is happening. So the alkyne group of EDU is exposed and it reacts with the fluorescent azide in the presence of copper catalyst. And then that's how you are getting the color finally, and you are staining the final product. You are doing this at room temperature. You need a reductant in this step as well. So as I mentioned before, click chemistry is a copper catalyzed reaction. So this is the click reaction happening here. You have the copper catalyst, you have the terminal alkyne, and you have the azide. So the azide and the alkyne, they're actually abiotic. They're inert until copper is added. So that's how you control any non-specific reaction. So just to give you an idea, what I mean by that is, like, so you are preparing your cell culture the day before. Next day, you add the DNA label. You label the DNA with a fluorescent azide. And then if you want to treat your cells with any inhibitors of DNA synthesis or proliferation, you can do that. You fix the cells, you permeabilize it, and then you perform the click reaction, which means you already, the DNA proliferation have happened, cell proliferation have happened, everything is done. DNA is labeled because you have added the EDU here. DNA is labeled. Now the click reaction is happening where you are adding the fluorescent azide, the fluorescent dye. You're adding the copper, and EDU is already there. And the final, the cells are already fixed at this stage, fixed and permeabilized. Final product, in the cells, you will have now the labeled cells. You are studying via microscopy or FACS analysis. So below is a diagram where I'm going to show you our data, where the panel 1, the panel A, where we have DIC is for labeling all the cells. So you can see there are cells. Azide, fluorescent azide, it's the dye. And so the red dye, you see it's all the DNA staining. with the EDU incorporated in it, it's in red. And total DNA, it's stained in green. So you can be sure that it's the replicating cells, is this proliferating cells, newly synthesized, the DNA synthesis that you see the DNA and the click reaction happening here. But in hydroxyurea, which blocks DNA replication, and this is in the HeRA cells, you see it blocks DNA replication, you don't see the click reaction here because EDU is not here, right? Because EDU is not incorporated since there was no DNA replication here. But this is the newly synthesized ones. But cells are here. And in the panel B, it's an example of a FACS analysis, flow cytometry, where we have used Jurkat cell, where you can see the green one is the proliferating Jurkat cells versus we have treated with ganciclovir, it suppresses DNA synthesis. So there is no, because there is no click reaction, so the peak is less. It suppresses the DNA biosynthesis. So this kind of tells you the specificity of the reaction. When you're suppressing DNA synthesis, you don't see that. But in the proliferating cells, you see that analysis of this newly synthesized DNA in the proliferating cells only. Okay, so now the next kit we have for click chemistry reaction based is for studying global protein synthesis, which is your K715, and the readout is via either FACS or via microscopy. What's the principle? So in general, proteins are generated by mRNAs, which is the ribosomes, and protein synthesis is not always proportional to mRNA because there is always degradation happens. So what's the classical method people use for studying protein synthesis? Is either incorporation of azide or alkyne methionine analogs. These are the two analogs of methionine. What's the disadvantage of the normal classical method that people have been using? That cells prefer methionine over these two, and you are using the analogs. So that the amount of incorporation of methionine will be less because cells usually prefer these two analogs compared to methionine. You cannot do animal studies, and you need to activate the tRNA in order for the protein synthesis. And this method, it generates total protein. You cannot study the nascent polypeptide chains. Unlike click chemistry, where you are able to study the newly synthesized polypeptide chains, not the final protein. So how does our click chemistry, our principle works? So here, as you know, pyromycin is an antibiotic that blocks protein synthesis. So in our click chemistry, instead, we use a pyromycin analog. So our kit is based on using this pyromycin analog, which is cell permeable. It contains an alkyne moiety. So remember, click reaction was alkyne moiety and azide. So this pyromycin analog, it contains an alkyne moiety and it's cell permeable. So this is a newly synthesized protein, nascent protein on ribosome and the tRNA. Since pyromycin mimics amino acetyl tRNA, so our pyromycin analog, it's going to replace the tRNA, and this is the new protein, as you see, that's NH2 primary amine group. So the tRNA is now replaced with our pyromycin analog. As you see here, the pyromycin analog attached to the protein, pyromycin analog, and with that alkyne group, right? And now this leads to premature termination because this analog is binding to the C terminus of the translating, this was the newly translating polypeptide chain, and pyromycin analog bind to it, the C terminus, and then that's how it stops the translation. So premature termination of protein synthesis. So then the reaction is the protein synthesis stopped. Now you are adding the fluorophore attached to the azide, and you have the alkyne already in the pure analog. You have the azide, you are adding copper as a catalyst, and you have this newly synthesized nascent premature protein, premature nascent polypeptide chain containing the fluorophore azide, because you had the fluorophore connected to the azide with the alkyne. Because of this alkyne, the fluorophore azide, and the entire product. So this is where the click reaction happened, and you have the final product. It has a fluorophore attached to it. Now you can study this final product via either fax or microscope. So what are the advantages of click chemistry? So as I mentioned, pyromycin is a cell permeable. It is readily taken up by the growing cells. You just add directly to the complete to the cell media. It's not an amino acid analog. So you don't have to depend on methionine analog, as I mentioned in the previous slide. So you just add directly to the cell will take it up, and protein synthesis will stop. It can be used with cell lines that are sensitive to media changes or incubation in methionine-free media. You can add even in presence of methionine, it doesn't matter. You can add this to your cells in a complete media. And now you detect the change in protein expression via this click reaction. Yeah, this is the modified protein, right? So you can detect this by either fax or microscopy. In the next slide, I'm going to show you some data. So panel A, it's a imaging. It's a immunofluorescence imaging. So the top panel, where it's the newly synthesized protein, which is labeled in red. We also labeled the DNA. Oh, hi, I don't know what happened. It looks like the computer stopped or something. Can you guys hear me okay? Okay. Yeah, not sure what happened. It stopped. So you all can hear me? Yeah, I want to make sure we are, because all of a sudden it stopped. So you all can hear me okay? If anyone can please type something so I know. Oh, okay, perfect. Thank you, thank you. Okay, I'll continue. What happened? Yeah, it skipped some slides. Oh, wait. Yeah, sorry, I don't know what happened. Okay, So on the panel B, we see the facts analysis of, facts analysis of, like, the negative control is your white. Positive control is all the labeled cells. Is it in red? And the cyclohexamide treated. So you see when the newly protein synthesized, you see label is more, versus when cyclohexamide treated, you see there is inhibition of protein synthesis. That's why you see lesser number of cells compared to your background, the negative control, which is not treated, not cells treated. Okay, so now coming to, so we have certain kits for studying RNA synthesis and translation. Based on click chemistry. So the classical method for studying RNA synthesis has been radiolabeling. So they use radioactive nucleotides, people use, which is very tedious. It's poor spatial resolution. Or they use tissue autoradiography, which is slow time exposure. It can take weeks or months. So the standard method, other than radiolabeling, commonly used is incorporation of BRDU, 5-bromouridine, or BR-UTP, bromouridine UTP. So what are the disadvantages? It is not cell permeable. Cell needs to be transfected with BR-UTP via liposome or osmotic shocks. It's expensive, it's time consuming, and it has limited applicability. And it's antibody-based method. What's the specialty of click chemistry of our kit? Is like it's cell permeable. And in this case, we are using another, it's analog, it's called EU, which is ethinyl uridine. So just like protein synthesis, we showed here, EU gets incorporated in the nascent RNA, but not in the DNA. So that kind of makes it specific. Instead, so we are not using uridine analog, but which is the natural uridine. Instead of using the natural uridine analog, we are using ethinyl uridine, which gets incorporated. And then the modified RNA you study via click chemistry. So you see a common pattern where it's always the azide reaction, azide with the alkyne group in presence of copper catalyst and a reductant. That's the typical, that's the cycloaddition click chemistry that we are using in all these kits. So just to give you an example, so here we are analyzing the modified RNA via click chemistry. So this is immunofluorescence. So you can detect either by microscopy or fax for these kits. So here you see the RNA label, but not in actinomycin D treated cells. So actinomycin D, it blocks transcription. How? By it binds to the RNA initiation complex, and that's how it prevents, the actinomycin D prevents transcription. So it binds to the DNA and the transcription initiation complex and prevents the elongation of the RNA chain. So as to know the specificity of the click chemistry reaction here, you see in the RNA label, you see that, but when you treat the cells with actinomycin D, you don't see the labeling. Although you see the DNA with the diapisteme, and this is the merge. So you can see the specificity for studying RNA biosynthesis using this kit. So the red represents the DNA was synthesized RNA as compared to the actinomycin D treated, and these are HeLa cells. Okay, so another kit which is also a very popular kit is for studying apoptosis, programmed cell death, or in situ DNA fragmentation using click chemistry. So this is the kit number. You can study the cells via fax or microscopy. The click chemistry principle for this kit, K191. Yeah, so what we do here is that it utilizes EDUTP nucleotide, which again gets incorporated at the 3' hydroxyl end of fragmented DNA. So it's by TDT, the terminal deoxynucleotide transferase enzyme, and it is detected using that same click reaction. This assay, it's easy to deliver because, as I said, so these are also cell permeable for incorporation of these nucleotides. You don't need anything fancy, you just add it directly to the cells. It's highly selective for detecting DNA damage, and the final readout is via FACS or fluorescence. And you detect a higher percentage of apoptotic cells than other antibody-based method where, DUTP is the normal tunnel assay that people normally use for antibody-based. So we do see a higher percentage of apoptotic cells. So the sensitivity is more. Yes, sensitivity for this kit is more using click chemistry based, and it's specific. So to give you an idea, how does it work? So panel A, we have shown the microscopic analysis of green is the nuclear stain, red is tunnel positive. So orange, which is the merge of both, it shows the apoptotic cells. So this is how it's going to look. This is an example of HeLa cells versus Jurkat cells. So if this is apoptotic cells where you see the tunnel positive cells. In the panel B is the FACS analysis. So that's how the analysis is going to look. Unstained cells, it's white. You have the background cells, control cells, where you have the click reaction, and then you have the negative control is this blue ones, which is the untreated cells. Tunnel click reaction, these are negative. And these are the DNase treated cells, which is after the click reaction, you treat the cells with the DNase. So as you can see, the peaks are very distinct. So this shows the selectivity of click reaction. So you are looking for the apoptotic DNA strand breaks. So you see all these merged ones, the orange ones, these are the tunnel positive cells, like both apoptotic cells. In the, it has the DNA as well, so. Okay, so another range of product in this click chemistry based is the phospholipid for assaying phospholipids. Let's see. So phospholipid assay kits. So we use this for if you want to study the global phospholipid synthesis. So as we know that phospholipid is a major component of this lipid bilayer, the phospholipid bilayer. And among all the phospholipids, it's the choline, phosphatidylcholine. It's the most common phospholipid in among other like phosphatidylserine or other phospholipids. So we use in this range of kits, we mainly use a choline analog. Phospholipid, it plays an important role, both regulatory roles in cell metabolism and signaling, structural as well as regulatory roles. And dynamics of choline containing phospholipid is essential parameter to analyze the cellular response under both physiological and pathological conditions. And to study the global phospholipid synthesis, we are using phosphocholine as the major, since that's the major, that's the major phospholipid. We're going to use that analog to study the global phospholipid synthesis. Okay, so what's the assay principle? So the idea is to visualize the newly synthesized phospholipids in vivo, which means inside cells. What are we using here? As I mentioned before, that since choline is one of the most abundant phospholipid present, so we are using the cell permeable choline analog. It's called propargylcholine or PCHO. So it gets metabolically incorporated into all this choline containing phospholipids. And since this is the maximum, it's the most of the, I think it's more than 50% of the phospholipid content is choline. So that's how gets incorporated. And the beauty of it is when you add this analog, it does not alter the natural lipid composition of the membrane. And then, so this is a schematic diagram where you are adding this analog, and during the phospholipid synthesis that gets incorporated because it's analog, so it gets incorporated instead of the original choline. The analog gets incorporated and the synthesis stops, and then you fix the cells and all, and then you add the, so this is an alkyl group again. You're adding the fluorophore attached to the azide. You're adding the copper reaction. This is all after fixation, cell fixation. Yes, after everything is done, and then you are analyzing the fluorophore. So this is the modified phospholipid. It has a fluorophore attached to it, and then you're studying this. So that's how you're studying phospholipid synthesis. To give you an idea how we do the assays, like either fax or microscopy. So this is a panel where you see the phospholipid label only in red, and then we always label with DNA to make sure those are, those are the cells, and this is the merge, and this is just a higher magnification image. So you see all this choline containing phospholipids in red. And on the right panel is the fax analysis where you see these are the vehicle treated, these are the phospholipid label, and phospholipase D treated. So phospholipase D, it confirms that the fluorescence, you see the red one, it is coming from the labeling of phospholipids, because this is the negative control. This is the click reaction happened there, so you see the color. But when you add a phospholipase D, then the signal is gone. So which shows that this signal is indeed coming from phospholipid. So you see the specificity of the click reaction. Another range of products that we have for studying based on click chemistry is for studying cellular glycans. So what are glycans? Glycans are vital components of glycoproteins, glycolipids, proteoglycans in all domains of life. They are appealing targets for molecular imaging, but as you know, glycans, they're very inaccessible with conventional approaches. So glycans, unlike DNA and protein, they are not directly encoded in the genome, and they are not amenable to imaging techniques that rely on genetic reporters, so you can't use genetic reporters. So what's the classical method? They rely on molecular recognition with probe bearing lectins or antibodies, but they are not well studied in vivo imaging. Click chemistry, the beauty of it is you can study in vivo. So glycans, we are imaging by labeling using these chemical reporters, which is the alkyne and azide reaction, and using the fluorescent probes. So the technique, this technique, our click chemistry, will help in the visualization of glycans in living cells and animals. So all our kits we have shown only in cells though. So what are the analog we use for metabolic labeling of glycans? So we have this, we have actually 3 different kits for that. For labeling glucanac or sialic acid, or we have for fucosylated proteins or O-glucanac. These are the 3 difference. N-acetyl manosamine or N-acetyl glucosamine for glucanac, or N-acetyl galactosamine. So what's the analog we are using? So these are the different kits, like for K441. This is for detection of N-linked glycosylation of cell surface proteins, N-linked glycosylations. K560. This is for detection of O-GalNAc. And we have K714 for O-GalNAc glycosylated proteins. Yeah, modified glycosylated proteins. Yeah, so we have different fluorescence, fluorophore, and we have K592 as well. This is for fucosylated proteins. For suppose if you want to study any fucose modified proteins, fucosylated protein, so you use that. So for 441, like let me tell you what do we exactly use. So for 441, we use the analog. It's called, we used a modified manosamine precursor. Instead of the regular manosamine, we used a modified manosamine precursor. For 560, we used a modified galactosamine precursor. For K714, what do we use? We used a modified, again, a modified analog, a glucosamine precursor, a modified glucosamine precursor. And for K592, we use a modified fucosylated precursor. It's also called fucose analog. So as you can see, we are using all these analogs and these are readily cell permeable. So you just add in the cells and then let the reaction happen. So I'll go a little more details, like for the N-glycosylated for K441, which is for sialic acid, and we are using here the modified manosamine precursor. You directly add in the cells and it gets converted to sialic acid, and then it's transported to Golgi for glycan elaboration. So you are using that precursor, which directly get into the cells, and then you do the click reaction by adding the fluorescent dye, copper, and alkyne already is in the manosamine precursor. It's already incorporated in the final product. So it's a powerful method for imaging the localization, trafficking, and dynamics of glycans. So as I mentioned, so here the non-fixed. So once the reaction happens, once the manosamine precursor, after treating the cells, you have the final modified glycan, and then you fix the cells, permeabilize the cells. So this is an example that it's non-fixed, non-permeabilized. You see all the glycans on the surface. This is fixed and permeabilized. And as I mentioned that once the manosamine precursor is fed directly into the cells, it's converted to sialic acid and transported to Golgi. So here are a high resolution image where you see that the glycans are transported to the Golgi apparatus. So this shows how easy it is to study glycans using our click chemistry based reaction. Okay, the last batch of products that we have for the EZClick reaction based is that for studying all this palmitylation or myristylation protein. These are also modifications, like palmitylation occurs when fatty acids such as palmitic acid is covalently attached to cysteine. And this is a post-translational modification, like palmitylation, sterilization, or myristylation, fucosylation. These are all post-translational modification, and that affects the cellular trafficking, compartmentalization, or membrane tethering. So in case of palmitylation, what it uses is a palmitic acid, a modified palmitic acid, not the regular palmitic acid, but a cell permeable palmitic acid. You directly feed into the cells and that it gets incorporated into the proteins during or post-translational. And then you are studying the modified protein, modified palmitylated protein by FACS or imaging. Same thing for sterilization. You are using a modified alkenyl. So all this modified palmitic acid or modified stearic acid, they all have an alkyne group. So you are adding here in this case, you are adding the modified stearic acid directly into the cells. It gets incorporated into the protein during our post-translational, and then the modified stereoylated protein is then detected via FACS or imaging. Similarly, for myristylation, in this case, you are adding alkenyl myristic acid, which is again cell permeable. You are adding directly to the cells, and it gets incorporated into the proteins during or post-translation, and then you are adding the copper, the azide, fluorescent azide, and you're detecting the modified myristylated protein via FACS for quantitative analysis, FACS or microscopy. And the fucosylated, as I mentioned, you are using a modified fucose as the analog, which is again cell permeable, and you are then detecting the final product, which is the modified fucosylated protein via FACS or microscopy. So to just give an idea, to summarize about all these click chemistry kits, 3 words. It's fun, you see the color change, it's fast, and it's simple. You don't have to worry about side reaction, you don't have to worry about different temperature, but it's specific, it's sensitive, it's simple, it's fast. So what are the things you can study? You can study cell proliferation, you can analyze cell cycle, you can screen for anti-cancer compound and agents, you can evaluate biologically active genotoxic compound because you're treating here, right? You can add the treatment when you're adding the DNA label. You can add all these toxic compounds if you want to evaluate any biologically active or genotoxic compound, you can add the treatment. You can study DNA fragmentation during apoptosis in presence or absence of treatment. You can study DNA, phospholipid synthesis. You can study modified glycoproteins or other post-translation modifications. How does it work? As I mentioned before as well, you culture the cells the day before. Next day, you add the DNA label or your other treatment, whatever you want to do. You fix and permeabilize the cells. So here already, while the cells are growing, your either modified protein analog, the pure analog for protein synthesis, or the choline analog for phospholipid synthesis, or the DNA analog for your DNA synthesis gets incorporated while the cells are growing. Once you stop, the incubation time depends. In the subsequent slide, I will show you the time range. For different assays, there's a different time range. And then once the reaction is done, you stop the reaction and you fix and permeabilize the cells and perform the click reaction outside, which means once it's permeabilized, now you're just adding to the wells. You are adding so this already has the alkyl attached to it, alkyl group attached to the incorporated pure analog or the DNA analog or other analogs. So you just add the azide, the fluorescent azide. You add the copper to catalyze the reaction, and you add a reductant to reduce. Reductant is needed, like a reducing agent, actually, reducing agent. And then final output, you analyze by microscope or via FACS. It's simple. Okay, to kind of summarize, what are the kits we have? So these are the different kits we have. Technical specifications, we have K946 for DNA, 718 RNA, we have protein, 2 different kits we have. We have choline. So I think one is plate based, that's why there are 2 different kits. And I think one is red, one is green. Otherwise, mostly it's green. We have this phospholipids for studying phospholipids using choline as the label is PCHO, choline. We have for studying all the sialic acid or the modified glycoproteins, these kits. I tried to summarize the incubation time. It could be for like DNA synthesis, it's fast. It's 24 hours to 30 minutes. You can do 30 minutes, 24 hours, depends on how fast your cells are growing. For RNA, hour, here 30 minutes to 24 hours, depends on how you can do a time dependency. Phospholipid, we say 24 hours. For glycoproteins, we say 24 to 72 hours. Size of all the kits, 100 assays. You can use suspension cells or adherent cells. So it's applicable to both. Application based, these are either fluorescence or flow cytometry. The excitation for facts, we have the different range. And simple click reaction is you're adding the copper reagent, the fluorescent azide, and you need a reducing agent. Reaction time, only 30 minutes. After this incubation, all you need is 30 minutes of reaction. Advantages, it's non-radioactive, versatile, it's specific, sensitive, it's convenient, and it's cost-effective. And it's adaptable with most commonly available instrumentation. So either flow cytometry or you need a microscope. And some we have plate-based. I will show in the next slide. So we have some plate-based assay as well. These 3 plate-based assay. We have a plate-based assay for RNA. We have for protein, we have a plate-based assay. Same pure analog. And we have one for phospholipid assay as well. We have a plate base. So for these 3 assay kits, you need fluorescent plate reader, a spectrophotometer, as well as you do either fluorescence microscopy or you can read on a plate. And then these are also the other kits, either red on green. We have 2 options for all these 3 different kits for studying these post-translational modifications of proteins. Same click reaction. You need the azide, the copper, which is after fixing the cells. So cells are not growing anymore. Only after fixing the cells and permeabilizing, then you are performing the click reaction. So which at room temperature, you don't have to be concerned about temperatures. Okay, I try to summarize in terms of the different catalog, the target, and the application of these kits. Like this is for DNA, the protein, RNA, phospholipids, glycoproteins, or sialic acid, or fucosylated glycoproteins. And these are some other kits for protein, RNA, phospholipid, or post-translational modification of protein, plus for studying apoptosis. So yeah, so that's it for me today. So thank you so much for your time, and I'm happy to take any questions. So you can type in the Q&A session. You can type in there if you have any questions. So thank you so much everyone for attending the webinar. If you have any question, please let me know. You can type in I hope you can see the Q&A session. Yes, please. Yeah, please type your question. I'm still waiting for the question. I see, yeah, someone has a question. So can you please type it? No, there is, so the question is, although you said you use mild condition buffer, is there any damage to cell by copper catalysis? No. So cells are already fixed by then. Cells are already fixed and permeabilized. So you don't have to worry about any damage to the cell. So the morphology is maintained. Yeah, so that's the beauty of this click reaction is cells are already fixed by the time you're staining the cells. Or, I mean, yeah, you can do FACS analysis, you can fix it. but that's not going to affect the morphology of the cells. So yeah, once you perform the EZClick reaction, you can directly go for FACS analysis. So it's not going to affect. Yeah. Yeah, it gives you this click reaction. It's very selective. So to get background signal, you are going to avoid many background signal. The principle is pretty simple and can be applied over a wide range. So if anybody else has a question, please type in the Q&A session. Maybe I'll give 2 more minutes and see. I don't see anyone typing. So, oh yeah, sure. Yeah, I love questions. Yes. So the next question is: there is any effects in presence of sodium azide? No. As I said, so until the cells are alive, which means that you are treating the cells, and then you are fixing the cells. You're fixing the cells, permeabilizing the cells, and then you are performing the azide reaction. So azide comes much later. You are first incubating the cells with your analogs, and that is all normal cell media, so not affecting, like not harming the cell at all. And once the reaction is done, you stop the reaction, wash the cells, and then you are fixing the cells. Once the fixation, permeabilization is done, then you are doing the click reaction and the DNA stain. So the azide, the question is, I wonder if there are any effects in the presence of azide. So the answer to this is no, there will be no effect in the presence of azide, since you are performing the azide reaction after the incubation with the cells have been done and after the cells have been fixed and permeabilized. Does that answer your question? Okay, thank you. So yeah, so some of the selling points that you guys can use for the for our customers is that it's simple, fast, room temperature, and especially the glycoprotein assay. Studying glycoprotein is not easy, but with the click reaction, it's easy, and you can do all this in vivo, and you don't need any fancy instrument. And it's quantitative. When you do the FACS, it's quantitative as well. It's sensitive. Do you have any other question? Maybe I'll give another 2 minutes and see if somebody has any question. Please type it here. So after the webinar tomorrow, we will send you all the slides as well. We'll email you all the slides, and then if you have some question later, you are always welcome to email us the question, and we are happy to answer the question. Yeah, maybe 1 more minute and then because it's so hard to know if someone has a question or not. So I'll give maybe 1 more minute and see if you have any question, then I'm gonna log out. Okay. Thank you, everyone. It was nice explaining the click chemistry kits and present you the different kits under this category. This is a new, we have added since our last webinar was in, I think, 2017. Since then, we have added a lot of click kits in this category. And we are very excited about this entire array of products under this category. We have about 20 products under this. So I was happy to present to you, and thank you so much for your time, and you all have a great rest of the day. And we will send you the copy of the presentation, the slides, maybe tomorrow sometime. And thank you so much again. Okay, bye everyone.